This is the Professor, CBMJ PhD. For today's Friday, October 13th, 2023. It is 3:01 and 48 seconds, AM Eastern Standard Time. And today's research, microbial diversity. So let's come to it here. Doubling down on known protein families using a novel computational approach. Researchers confirm microbial diversity is wilder than ever. Date October 11th, 2023. Source: Lawrence Berkeley National Laboratory. A massive computational analysis of microbiome datasets has more than doubled the number of known protein families. This is the first time protein structures have been used to help characterize the vast array of microbial dark matter. Microbial communities living everywhere, from soils and stomachs to the deep sea, are capable of doing a lot of unique things when it comes to energy cycles, turning biomass into things like ethanol or hydrogen or solar energy into hydrogen. Microbial communities are also incredibly difficult to study. Many of the microbes within them cannot be cultivated in laboratory settings, since each microbial community has its own unique makeup of microbe players and the functions they perform. Artificially replicating a whole community is impossible. Metagenomic sequencing allows researchers to study the entire genetic makeup of these communities via whole genome sequencing of the samples without being able to distinguish which gene belongs to each individual microbial species within a community. Therefore, the process hinges on referencing to existing genome sequences. And some of these proteins are what the scientists call known knowns. That is, they're similar to genes, but known function. Others are called known unknowns. That is, they're similar to previously known genes from isolated organisms, but we still aren't sure of their function. However, if a gene in a community doesn't match any of the previously known genes from isolates, there isn't much scientists can tell about its function or its origin. As a result, these genes were typically discarded from any analysis as useless information. These represent the unknown unknowns because they aren't similar to anything they've already defined. A huge percentage, around 30 to 50% of the protein families that they do so far, still does not have any known function, but they do the families. Yet almost 20 years of metagenomic data and metagenomic analysis, and still there has been no real analysis of protein families from metagenomes per se. Recently, other research teams have leveraged the power of artificial intelligence to decode the language of protein sequences and obtain hints of their possible functions. Yet these efforts were limited to the realm of already known protein sequences. In this endeavor, they have not only ventured into the uncharted territory of understanding the vast landscapes of functional diversity, but they have also pushed the boundaries by applying artificial intelligence methodologies to unravel their roles. And consequently, they have amassed an extensive repository of groundbreaking insights, significantly expanding the horizons of potential functions across various categories of proteins, including those with pivotal applications in biotechnology, such as DNA editing enzymes. The discovery of new protein families has started to plateau in recent years, perhaps suggesting that scientists had captured much of the diversity out there, even if it hadn't yet defined what it did exactly, but what kind of diversity might those unknown unknowns hold. The team started with 8 billion petagenome genes from IMG. The study also references data from the JGI's genomes from Earth's microbiome, or GEM, catalog that then removed any genes with even a remote similarity to previously known genes, leaving them with around 1.2 billion novel genes. They took what they were left with and clustered them into families. From there they focused on families with at least 100 members. If you have 100 sequences, the quality of the cluster is significantly higher because it is very hard to have 100 sequences from different locations or habitats that align very well randomly. Replicating that 100 times would have been almost impossible. When the team was finished with this phase, they found that the protein family diversity within this metagenomic space, the unknown unknowns, was vastly greater than that of the reference genomes by at least double. As they keep on adding more samples, they are getting more protein families. In a few years, as they keep on sequencing more metagenomes, some of the clusters that have currently 50 members or more will grow to 100 members or more as well. So, they are saying diversity has doubled, but in reality, it could be 3 or 4 or 5 or tenfold more out there. While the team didn't drill down, they were able to further characterize these families. They divided the protein families up by environment and found only 7% of protein families were shared across all 8 environmental categories. Instead, families preferred a specific environment, whether that be soil, animal hosts, marine ecosystems, etc. And so they must be doing something interesting or important for that habitat. That is definitely material that the scientific community now can use better. Let's say somebody is working on soil environments for the human body. They may take some of those families and try to functionally characterize them because they are very specific to that habitat. A taxonomic analysis found that the majority of these protein families belong to bacteria and viruses, though 6 million of the sequences evaded classification. The researchers also tried to hone in on the function of the genes via 3D modeling and comparing structures of the unknown to those of the known. Similar structure equates to the high likelihood of similar function. The team also identified protein families with completely novel structures. And this is the first time protein structures have been used to help characterize the vast array of microbial dark matter. The study took roughly 2 years to complete, with only about 20,000 metagenomes sequenced at the time. Now that the number is closer to 60,000. There is still 70 to 80% of known microbial diversity out there that is not yet captured genomically, so that diversity is definitely holding a lot of new secrets in terms of functional diversity as well. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.